DNA is in everything. DNA is super important because it's the genetic code for life. But what if I told you we can alter the DNA of other organisms?
Genetic engineering is the deliberate modification of the characteristics of an organism by manipulating its genetic material. When people think of genetic engineering, they think of scary things like Dr. Kurt Connors from The Amazing Spider-Man turning himself into a lizard, which is pretty cool if you ask me. But genetically modified organisms can be so beneficial for our health and the environment. Just think of crops that have been genetically engineered to be pest resistant so we don't have to ingest dangerous pesticides. Genetic engineering is such a huge field, but today we're going to narrow it down and talk about genetically engineered plants. We're going to use the example I talked about earlier with pest-resistant crops. But first, let's get some background info. The hydrogen bonds that keep together the base pairs of DNA, adenine, thymine, guanine, and cytosine, are super weak. This allows them to be broken easily to extract DNA from the donor organism and allow for the insertion of new genes. For genetically engineered plants, the most common bacteria used is Agrobacterium tumefaciens. But to make our crop pest resistant, we will use the bacteria Bt. Bt is a bacterium that occurs naturally in soil. Strains of Bt produce proteins that kill certain insects. When insects ingest Bt, it disrupts their digestive system and ultimately leads to death. Bt is very selective, meaning different strains kill different insects and only those insects. Geneticists can engineer plants to make their own Bt to target certain insects. Geneticists take the isolated gene from the chosen Bt strand and input it into the plant cell. To help break down this process, we'll summarize it into 4 steps. Number 1. Restriction enzymes cut the chosen and isolated gene from the DNA, in this case the Bt. They then extract the gene that prevents the targeted insect from the DNA. Number 2. The same restriction enzymes cut the same shape as the chosen gene into the DNA plasmid of the plant. Because the same restriction enzymes are used, the sticky substance that they leave behind is complementary to the gene we cut. This allows for the joining of the chosen gene in our DNA. Number 3. An enzyme called ligase is then used to finish joining together the gene and the DNA. Number 4. This newly modified DNA is then inserted into the plant cells. Researchers grow modified plant cells in the presence of antibiotic or herbicide. In a tissue culture, they select the plant cells that are unaffected and grow them into full-grown plants. These plants are classified as transgenic organisms, which is basically a fancy term for genetically modified organism. These new plants then produce the lethal pest protein that is present in the Bt bacterium. This foundation of genetic engineering can be used for so many things. Although the type of bacteria and the DNA of the organism may change, we can use this process to genetically modify plants to have a longer shelf life, to produce more yield, or to possess vitamins that weren't present before. So the next time you see that GMO sticker on the produce you're buying at the store, you know the process that brought it to you.